Good afternoon, everyone. I would like first to thank the organizer for inviting me today, and especially Carrie. She was very nice. Thank you for the introduction. And I'm really excited today to be here to presenting my work, and I'm truly honored to be here with this lineup of incredible speakers. So the study that I'm going to present today is new data on memory implantation in absence of experience. So every action that we make leads to the activation of specific neurons in our brain. And even now that I'm speaking to you, my brain is encoding special information about this new room and the new faces that I'm looking at. And in the same way, when a mouse is performing a behavioral task, specific neurons in its brain are active. And the research for codes in the brain that encodes information due to external stimuli is one of the most challenging fields in neuroscience. So revealed code in the brain are, for example, cells that are active when a mouse is in a specific location, like the place cells, or cells that are active to encode visual information, taste information, or odor information. So during a Pavlovian fear conditioning, a mouse learned to associate a neutral stimulus, a conditioned stimulus, like an odor or a context, with an aversive stimulus, like a series of food shock. And this will lead to the formation of a conditioned response for the originally neutral stimulus. In this case, the mouse, for example, will spend less time in the compartment in which the conditioned odor is presented. And in particular for this task, as you can see, only the mice in which the odor is associated with the shock present the mice spend lower time in the compartment room in which of the conditioned odor. In fact, the rest of the control group doesn't present this behavior. They will spend the same time in the two compartments. In particular, naive mice, or mice in which it's presented, we presented only a series of food shock, or mice that are presented only with the odor. And in particular, the most important control experiment are these mice in which the two stimuli, the odor and the shock, are presented delayed by 24 hours. And this means that the mice cannot actually associate the two stimuli. So when the mice are learning this specific task, specific cells in their brain are activated. Specific codes in their brain are activated. But what's the rule of these cells? What's the rule of these codes? So in the past decade, using different technique, many papers have been published to show that it's possible actually to tag these cells, to selectively tag these cells. For example, when a mouse is performing, learning a contextual fear conditioning, in which he has to associate a series of food shock with a context, and it's possible to tag these cells. And then when the mouse is put back in the context, in the conditioned context, and these cells are inactivated in different ways, the mouse can't remember the behavioral task. So the introduction of optogenetic has been of terrific relevance for this study. In fact, before optogenetic, it was impossible to casually link the real-time contribution of subpopulation of cells in cognitive tasks. But now with optogenetic, we can actually tag subpopulation of neurons and optically activate them while the mouse is performing a behavioral task. So in my experiment, what I wanted to answer is a very simple question. Does the code exist? And to really answer this question, we should be able to induce the formation of a memory of an event never experienced before, only substituting the external stimuli with the code that is like encoding the external stimuli in the brain. So to do this, we need to know. We need to use a well-known code in the brain. And when we look at place cells, we can't actually tell a priori which cells will be activated when we experience a specific place. So, for example, if we go to Google Maps and search for a specific place in the world, like Toronto, Rome, or the Hawaii, we can't actually know which cells will be activated in the hippocampus. But there is a specific system in our brain, that is the olfactory system, that has this unicity in which for each odor, you can actually create each odor creates a unique map of activation in the olfactory glomeruli. In fact, in this glomerular activity response archive, this is a website in which you can search for a specific odor, like acetophenone, methylsalicylate, or carbon, and you can look at the map of activation in the olfactory bulb that these odors will lead. And this is possible because every time that the mice smell a specific odor, specific olfactory sensory neurons will be activated. And even if the olfactory sensory neurons in the olfactory epithelium are millions, each of them will express one and only one subtype of the olfactory sensory receptor, of the 1,000 olfactory sensory receptors that are present in the mice. And each olfactory sensory neuron presenting the same receptor will send their projection to one and only one glomerulus in the olfactory bulb. And this, the position of this glomeruli is conserved across mice. So this means that every time that the mice will smell a specific odor, we will know for sure the activation of the glomeruli that this odor will lead in the olfactory bulb. In particular, for example, the acetophenone is an odor that activates a specific subpopulation of glomeruli, including the M72 glomerulus. The carbon, instead, is another odor that will activate a different subpopulation of glomeruli, not including the M72. So in my experiment, what I wanted to do is actually substitute the experience of the odor with the stimulation of the glomerulus, the M72 glomerulus in this case. And I will do that during the presentation of a series of food shock. Then I will test the mice in an odor preference test in which they can choose to be in one of the two compartments, each of those compartments showing a different odor, presenting a different odor. And the two odors that I will present are the acetophenone, that is an odor that activates the M72 glomerulus, or the carbon, that is actually an odor that doesn't activate the M72 glomerulus. So if this is true, if it's true that we can actually substitute the odor with the stimulation of this code in the brain, we should be able to see an avoidance for the acetophenone odor. So to do this, I used an optogenetic approach, and I've used the M72 mice, in which the expression of the channel rhodopsin and YFP gene is under the control of the M72 promoter. And here you can see an example in the olfactory glomeruli of these mice, in which the M72 glomerulus is the only one expressing the YFP gene, while all the other glomeruli surrounded are not. So in this experiment, what I did was to pair M72 stimulation with a series of food shocks. And in particular, the stimulation that I've used mimics specifically the activation of the glomeruli that an odor will lead to. And in particular, the pulse of this stimulation will be mimicking the smelling period of the mice of 100 milliseconds. So What I did here was actually to stimulate the M72, pairing it with a series of food shock. And as you can see, the mice during all the 10 minutes of test showed avoidance for the acetophenone odor. And here there is an example of these mice in which, in which the association between M72 and the shock occurred. And you can, as you can see, the mice actually avoid the acetophenone compartment. And for this experiment, I ran all possible control just to show that the M72 stimulation alone can't actually lead to the formation of an avoidance or a preference for the acetophenone odor, as well as in mice in which the M72 stimulation and the delivery of the shock are delayed by 24 hours. So the mice can't actually associate the two stimuli. And here there are all the experiments that I've showed you before summarized. And as you can see, all the control group doesn't prefer any of the two odors, but only in the case of mice in which the M72 stimulation plus shock occurred, or in mice in which the real odor is associated with the shock, we have an avoidance, a very similar avoidance for the acetophenone compartment room. So I also showed that if we train the mice for M72 stimulation plus shock, and then we test them later on, 30 days after, we have the same results. So it seems that this effect is also long-lasting. So in the last part of the experiments that I did, I really wanted to show that it's possible to completely substitute the external stimuli, just stimulating the codes in the brain that are encoding for this external cue. And in particular, what I want to do is not only substitute the odor here in the brain, but also I want to substitute the experience of the shock with a different code. And to do this, I've used this approach from Lammel, from the Lammel paper in Malenka's lab, in which they show that there is an input-specific control of Rewardin aversion in the ventral tegmental area. In particular, due to different synaptic connections between these two regions, the lateral bandula send projections to the VDA, and these projections lead to avoidance behavior in mice, while the lateral dorsal tegmentum send projections to the VTA that seems to lead to reward behavior in mice. So what I did here was to inject an adeno-associate virus containing channelrhodopsin in the lateral bandula. And here you can appreciate the projections going to the VTA. And what I did was to put an optrode right above the VTA to specifically activate these projections going from the lateral bandula to the VTA. And in the same time, I also stimulated the M72 glomerulus. So in home cage mice, I stimulated these two codes in the brain, the M72 glomerulus and the projections going from the lateral bandula to the VTA. And then I tested mice again in our odor preference test. So what I showed here is that the mice that experienced the stimulation of these two, M72 glomerulus and LHB, showed avoidance, also in this case, of the acetophenone odor. And here there is only the control group in which the two stimuli, the M72 glomerulus and the LHB VTA, are delayed by 1 day. So the mice can't actually associate the two stimuli. And what I really wanted to do, was to induce a reward behavior for these experiments. So what I did was, in this case, to inject a virus, the AAV virus containing channelrhodopsin in lateral dorsal tegmentum. And I specifically targeted the projections going to the VTA. So I optogenetically activate these projections while I was stimulating the M72 glomerulus. And here in the right panel in green, you can see the behavioral results. So you can see that in this case, the mice are actually preferring the acetophenone compartment room related to control mice in which the two stimulations occurred in 2 different days. So in this case, it's for the first time I found that we can actually induce the preference for this acetophenone odor. So to summarize this behavioral experiment, what I found is that it doesn't matter if we actually teach a mouse to associate a real odor with a real shock, or if we actually stimulate the 2 codes in the brain that are involved in these 2 processes, because the mouse will always avoid the acetophenone odor. And in this case, they will avoid an odor that was never experienced before. So we know that each kind of learnings lead to plastic changes in our brain. And these occur at different levels, from a synaptic level to a network level. And in particular, when we recall the memory trace that was learned, we have a reactivation of specific regions in the brain, a specific subpopulation of regions in the brain. So what I wanted to verify here is if the real memory and the artificial memory that I induced with the stimulation of this, stimulation of M72 and the VTA projections lead to engage to similar circuits in the brain. And to do this, I used the CFOS analysis, and I analyzed different brain regions, regions that are known to be involved in the olfactory processes, regions that are known to be involved in the aversion processes, and also regions like the amygdala that are known to be involved in the association between conditioned stimulus and unconditioned stimulus. And to do this, I trained mice for a real memory. Mice to associate an odor with the shock, and then I tested the mice only presenting the odor, the acetophenone odor. And as control group, I have mice in which only the odor was presented during the training, and then 1 day after they were tested again for the odor. To analyze the artificial memory instead, what I did was to stimulate the M72 glomerulus together with the projections going from the lateral amygdala to the VTA during the training session. And 1 day later, I only stimulated the M72 glomerulus to induce the recall of the memory trace. And as a control here, you can see mice that are stimulated only for the M72 glomerulus, and then tested again with the stimulation of the M72 glomerulus. And here you can see some slices of this analysis showing all the regions that I've analyzed. And today, I'm going to show you only preliminary data for the basolateral amygdala. Actually, the basolateral amygdala, as shown from KTI, is a very important region for the conversion of conditioned stimulus and unconditioned stimulus. And here there are some examples of the experimental group that I've used, naive mice, or mice that are trained only for the presentation of the odor, or only for the stimulation of the M72, or mice in which the pairing of the odor and the shock, or the pairing of the odor and the M72 glomerulus and the LHB are presented. And here there are some examples of CFOS expression in these different groups. And here there are the results for the CFOS analysis. And as you can see, both memories share the similar activation of CFOS in these 3 groups. So as you can see, only in the group in which the 2 stimulations occurred, there is an increase of CFOS expression, showing that probably these 2 memories also share the similar circuits. So gradually, we are leading to realize that the universe that we know and that we experience is indeed a representation of our brain rather than merely the world outside us. And during the last Canadian Association for Neuroscience meeting, John O'Keefe concluded his talk speaking about future of neuroscience. And he said that if we really think that there is a code in the brain, then we can use it to implant false perceptions, emotional memories. And I was in the audience, and I get really excited because that's exactly what I've showed you today. So it is possible, if we know the code in the brain, to induce the formation of artificial memories. And with this, I want to thank Paul Frankland and Sheena Jocelyn, in which I'm doing my postdoc, and Sarah Moberg, the students that work with me at this project, as well as Leonardo Restivo, and all the JEF lab. And also, I want to thank the collaboration that we have for this project with Kerry Ressler and his student, Philomena Morris. And here there is, like, as shown from Sheena before, all our group. And in particular, I want to show you how in the JEF lab we love to create false memories. Since also in the Engram Social at the SFN, we were able to do it.